All right, let's go ahead and get started. So once again, thank you so much for joining Synthego here for our webinar on industrialized CRISPR cells powered by the Eclipse platform, where we're saying that you don't need to leave any variant or gene or mutation ever unstudied again. Okay, so today what we're going to cover in this webinar is an introduction to our genome engineering platforms, and also how our Eclipse platform that we've developed will help to accelerate disease research. And so Jason Steiner will be covering those first two topics. We're then going to do a deeper dive into the actual Eclipse platform, its capabilities that we feel really makes it the new gold standard in generating CRISPR cell models. And that's going to be followed by how this industrialized CRISPR process using, in this case study, iPSCs, induced pluripotent stem cells generated by Synthego can enable large-scale research projects. As an example, a large project we worked on for the National Institutes of Health. And then we'll be closing out with some Q&A. Okay, so you've already met me. I'm Kevin Holden. I'm the head of science at Synthego. I'll be your host today and leading us through the webinar. Joining me will be Jason Steiner, who is the Chief Strategy Officer at Synthego, and he'll be going over the platform overviews at the beginning. And then taking on the majority of our webinar today will be our Director of the Eclipse platform, Grace Thamaraja. Okay, so just as a quick introduction to Synthego and who we are, we're a genome engineering company. We really believe that it's possible for everyone to be able to access this new transformative technology of genome engineering utilizing the CRISPR-Cas9 technology. And so at Synthego, we really believe that you can leverage machine learning, automation, and this technology to build platforms for doing science at scale. So we're really trying to enable the acceleration of life science research through our development processes and these platforms that we've built, really with the goal of trying to improve human health through building the right kind of models and developing the right kind of insights for your research. Okay, so I'd like to hand it over now to Jason Steiner, who is going to talk us through the industrialized CRISPR platform that we've built around our genome engineering capabilities.
Thanks, Kev. And thanks everyone on the line for attending. As Kevin said, my name is Jason Steiner. I'm the Chief Strategy Officer here at Synthego. Before we jump into some of the main content around the Eclipse platform and the industrialization, as we'll talk about it, of CRISPR-based genome engineering, I want to take a few minutes to frame where the industry is at and some of the confluence of different technologies that are enabling a new way of looking at biology at scale. So we're all likely familiar with this image. It's become rather famous, mostly because of how extraordinary it is. The cost of sequencing a base of DNA has dropped about 5 orders of magnitude over the last 2 decades, the majority of it since the advent of next-gen sequencing. And really the amount of genomic information that we have sequenced is truly extraordinary. This information has been studied, analyzed, correlated, cross-referenced with phenotypic data, clinical data, and other data sources in thousands of genome-wide association studies in an attempt to really identify what the underlying drivers of certain types of biology, particular disease, are. But we haven't really reached the level of expectation that we had anticipated, the level of understanding. There have been a number of publications describing this around precision medicine, personalized medicine, genomic medicine, and so forth, and we're not quite there. And it perhaps is best summed up by this quote in the bottom right from the article in the Journal of Clinical Investigation. What we have are signals that we can't track back to causal variants. And when we can't track signals back to causal variants, we have little biology from those signals. So in short, we've been reading a tremendous amount of genomic information for many years, but we haven't really had the tools to really deconvolve it. And biology is a very complex network. To study complex networks like biology, One of the most effective ways to do so is to be able to perturb or modify the nodes of the network at a scale and with a precision that is comparable to the size of that network, and then to be able to measure and evaluate what the outcomes are. So genome engineering technologies, particularly genome engineering technologies like CRISPR, are beginning to enable this complementary scale to what we have been sequencing for the last 20 years or so. So this is an image that may be familiar to many. It's the Cas9 CRISPR system showing the nuclease, Cas9, the target DNA, and really the flexible component or the guide RNA, which is the programmable component. CRISPR is pretty unique as a genome engineering system in that it genuinely is a programmable system. And when we're talking about programmable, essentially in a similar sense as software, you can actually code the sequence that you want to modify. And you can do so in a sufficiently scalable way simply by printing, in this case, many, many guide RNAs. So this allows you to essentially arbitrarily address any part of the genome, and as a consequence, arbitrarily address any part of a biological network at a scale that is truly enabling and is the complement to the work that we've been sequencing. But I would say that in CRISPR is still actually not a particularly easy technology. It is a co-opted microbial immune system. It is still complex and a bit stochastic, and so optimizing it and doing it effectively is still a challenge, despite its democratization and ease of perceived use. Many researchers are really not achieving the results they desire, and the effort itself is relatively laborious. We performed a large industry study last year, and these are some of the results of the study. At a high level, we found that many researchers indicate that they can take as many as 7 tries to get an effective CRISPR edit. It can take up to 100 hours to make a single cell model. And as a consequence, many researchers are studying less than half of the genes that they'd like to, and potentially many more genes than would be necessary to truly understand and deconvolve disease. So despite the programmability of CRISPR as a system and the flexibility, right now it is not necessarily approaching the scale that's necessary to really understand disease, but it needs to. To begin to really unlock the complexity, CRISPR needs to be vastly scalable as a system. And we think about that as moving it from essentially a science experiment into an engineering protocol. Some of the terms that we use are we need to industrialize it, we need to standardize it, it needs to be scalable, and it ultimately needs to be predictable and have all of the features of engineering disciplines. So we're at the intersection right now of a number of technologies, both inside the field of biology and out, that are actually creating this possibility. So CRISPR as a programmable gene editing system is one of them, combined with high throughput automation technologies for experimentation and machine learning are actually driving a lot of this standardization. And at Synthego, we are building CRISPR-based high throughput manufacturing platforms to implement these types of principles. So these platforms, we have 2 of them. We will discuss one of them in detail here. And the other one, Halo, is our platform for generating CRISPR engineering tools. So these are tools for actually doing CRISPR at scale. And Eclipse is our platform for generating CRISPR engineered cells at scale. What's important to note is that the platforms that we are building are more than automation hardware. If we extend the analogy of CRISPR as a program or a programmable type language, our platforms are more akin to an operating system, like a computer operating system. They are full stack assemblies of a variety of different technologies, including proprietary high throughput hardware and software that we have built, bioinformatics and closed loop machine learning protocols, and proprietary chemistries and protocols in science that we have developed in-house. So these capabilities are integrated together into 2 primary platforms, as I mentioned, Halo for genome engineering tools and Eclipse for genome engineered cells. And the combination of these 2 platforms enables a whole host of products that support applications from basic discovery biology through scalable cell manufacturing. One of the real important things about these platforms and the integration of these manufacturing platforms is that they are constantly improving. So as I mentioned, CRISPR, like most biological processes, is stochastic, and there is some degree of unpredictability in it, but it can be learned and it can be modeled through this type of iterative machine learning protocols. So we have a protocol that we implement across the company on all of our platforms, which we call closed loop CRISPR. It links our platforms for generating engineered tools and our platform for generating engineered cells. Our tools platforms provide reagents to engineer cells. Our engineered cells outputs provide data back to our tools platform. And in that process, in that closed loop process, we continually learn and improve. And so what we find is that over time, as we do more and more work, we actually continue to improve our efficiencies. We improve the scale, the predictability, and the throughput of the work of the products that we produce. And this is an iterative process. It continues to get better the more that we do as we turn CRISPR, again, from a scientific experiment into much more of an engineering protocol. A very brief snapshot of some of the underlying features and technologies that support our two platforms. So our Halo platform is composed of proprietary hardware, software protocols, and chemistries. These produce high-quality genome editing tools as products that support researchers across the spectrum from basic discovery through therapeutic reagent manufacturing, including a GMP guide RNA facility that we have built. We do have closed loop feedback on this as well that continually improves the quality and the design, particularly the bioinformatic design of our products. And we have a wide range of production scales that supports synthesis requirements for many applications. The Halo platform here, as I mentioned, is vertically integrated with the Eclipse platform. I won't spend too much time on this slide because Grace will cover it in detail later, but the two platforms are related and they learn from each other. By combining these two platforms, we're able to produce a whole suite of products, and these products support the utilization of genome engineering technologies across the entire discovery to clinical manufacturing spectrum, including everything from target identification through, again, GMP manufacturing. So we have both genome engineering tools and we have a suite of genome engineered cells for things like target discovery, scalable disease modeling, GMP manufacturing, with the goals of really accelerating the transition and the process of discovery research all the way through clinical introduction. So we provide a continuum of products across the spectrum. As we are continually improving our platforms, it actually allows customers and partners to ask larger questions and get faster insights. And this type of scale that's enabled by this continuous learning and the industrialization of CRISPR genome engineering is actually practical and meaningful. So this is a summary of a collaboration that we had done earlier last year. We engaged with a broad-based collaboration to address COVID. And in an application of the speed and the scale that our platforms have enabled, we partnered to translate a protein interactome, which is the protein-protein interaction network between, in this case, SARS-CoV-2 and the human proteome, into over 300 cell models that we produced in just a few weeks. These models were then used in further studies as the basis for identifying 66 putative targets and eventually having a candidate in the clinic within 10 months. So there were publications associated with each of these steps. But really, this is the meaningful application of the type of translational scale that can be enabled and the types of questions and challenges that can be addressed through the ability to do highly predictable, highly scalable CRISPR-based genome engineering. So these types of platforms are really needed to match the scale and the scope of the vast amount of information that we have been collecting the last few decades, and really to accelerate the understanding of biology and the drivers of human disease. And what's really quite exciting about this is being able to do this genome engineering at scale and translate that information back into cell models in a highly predictable and highly scalable way. Combining that with other technologies like high throughput phenotypic analysis and the rise of a whole class of machine learning technologies is really potentially going to be able to allow us to unlock a deeper understanding of disease, studying not one or two, but potentially hundreds or thousands of genes at a time and their interactions to accelerate the development of novel therapeutics. So that's kind of a description of the platforms that we're building. And I'll pass it back to Kevin now for the transition to discuss one of these platforms, our Eclipse platform, in more detail.
Okay, thanks, Jason, for that very in-depth discussion about our platforms at Synthego. So it's my pleasure to introduce Grace Thamarja. And Grace is going to take us through a deeper dive into the technical capabilities of the Eclipse platform. So thank you, Grace.
Thanks so much, Kevin, for the kind introduction. So I'll begin by saying that Jason articulated the undeniable value that CRISPR provides as a research tool to develop cellular models, right? However, up until recently, every time CRISPR was used to generate an engineered cell, it felt more like an experimental process. And so what the platform that we've developed at Synthego allows us to do is it allows us to move away from that experimental feel and towards a more predictable outcome for editing. And so whether you're doing hundreds, as Jason shared in his case study, or just a handful of edits, the platform will be of benefit to you. And so we'll take you through now a little bit more of a detailed look into the Eclipse workflow and how the platform enables better editing over time using real-time data. Jason highlighted a few of this in a slide that he passed through. I'm going to dive really deep into it. But before I get into the Eclipse aspect of it, let me highlight at least the Halo platform, which sits upstream of our Eclipse platform. So Halo generates the gene editing tools that are used in the Eclipse workflow. The majority of what I'll be talking about downstream now will focus specifically on Eclipse, but I just wanted to highlight where Halo fits into Eclipse there. So the strengths of these platform of the Eclipse platform are highlighted here with the five other squares rather that you're seeing, and we'll go deeper into each of those capabilities, sharing data from the platform along the way. So we'll start with the top three that you're seeing, really focusing on the modular design and how that modular design allows us the foundation that we need to generate to make quick changes and make improvements as they're needed. So let me just spend a quick moment on the modular design. So it's really the foundation of our platform, and what it allows us to do is quickly pivot. And so we can make changes quickly, as I mentioned, and easily. And the changes that we make specifically increase capacity. As we mentioned, we want to have that scalability option here. And also it allows us to make improvements that allow for the generation of new products or features to be added very easily. So when we look a little bit more clearly on that, the details of the modular workflow can be summarized along what you're seeing at the top. They've been bucketed into these larger categories, such as feasibility, intake and design, factory, and fulfillment. And so feasibility refers to where our editing strategy is developed. Intake and design is where the cells and reagents are ordered and they're prepared for factory use. And the factory is where the actual editing happens, which also includes the clonal isolation portion. And then fulfillment is the generation of the products, aka the cells, that will be prepared for shipment and passed on to the researcher for further assessment and downstream assays. Of course, you can imagine that the transfection module is a very pertinent one. So we're going to spend some time on that, and we'll also spend some time on the clonal isolation workflow as well. And so let's dive right into the transfection, the optimized transfection that is captured in our module. So our high throughput method uses used for transfection allows us to test several parameters at once. In this way, we can actually result out hundreds of conditions in a very short period of time. These results, as Jason mentioned before, help us learn and design better the subsequent edits. And so the data, the volume of data we're getting really allow us to make good decisions as we move forward and in a predictable way. And so let's spend a little bit of time looking at that workflow. So the automation doesn't just allow us to do these countless edits, these countless transfection conditions at once, as I mentioned, but it does a good job of moving the edited cells in a parallel way through the various processes that are downstream of transfection. So if you follow me at the top, what you can see is at the same time that we want to, so at the same time that we want to move on to the clonal isolation workflow, which is at the bottom, we're also able to generate cells to confirm genotype, as well as generate the retains and carry out some QC testing that will make the final product at the end of our as the cells make their way through the platform. And so let's spend a little bit of time on how this works. So as I mentioned, we do all of these things simultaneously, which is one of the many examples that pull out efficiency in our workflow. And so as we're genotyping the cells that come off of the transfection work cell, we also are expanding them and getting them ready for clonal isolation. So once the genotyping data comes through, we can now move very seamlessly onto the clonal isolation portion. And at the same time, as I mentioned, we're managing retains and ensuring that we can go back to a part of the workflow in case we need to in the future. So what does that look like in terms of data, right? So the amount of data that we're generating through this transfection process is absolutely powerful and has considerably informed our decisions. And so here's an interesting example that I want to share with you of a data set that shows the impact of cell line on editing efficiency. So we show 3 cell lines here. So EMT6, which is a mammary carcinoma line, 5637, which is a bladder carcinoma line, and Kasumi3, which is an AML or leukemic cell line. And so what we have here is over 200 transfection conditions that are carried out or performed on each of these cell lines. And we have a nice representation of these cell lines being either easy to edit, therefore having high editing efficiency, or more challenging. And so let's take a little bit of a closer look on this. So a single guide was used to generate the values that you're seeing here for the editing efficiencies. It's associated with a knockout. As you can see on your far left, EMT6 is a fairly easy cell to edit. So the majority of the transfection conditions that we used worked well. And in contrast, Kasumi3 is a more challenging cell line to edit using that same guide. And so with Kasumi3, what we see is a very small fraction on your far right, a very small fraction have editing efficiencies that are greater than 50%. And then of course, the 5637 cells fall somewhere in the middle, with about half of the transfection conditions yielding editing efficiencies that are greater than 50%. And so given the scalability of and this is an example of 3 cell lines, right? But given the scalability that we've built into our platform, we can easily estimate the ideal transfection condition for many cell lines. And we can do this as we onboard these cells onto our platform. And so as a result, we've actually been able to optimize transfection conditions in more than 300 cell lines across more than 15 tissue types. And so here we are showing along the bottom of interest to you we're highlighting the highest editing efficiency obtained using a single guide across several cell lines. And then we just quickly highlight the tissue type that those are linked to. And so what you can really see here is, the ability of our platform to do this in a very easy manner, which makes it ideal for that scalability part, but also for learnings, because we can learn from all of these edits that we do fairly quickly. And so we spent some time on transfection. In addition to transfection, I think another important module in our workflow is clonal isolation. So that's been highlighted at the bottom there. So we're turning back to that slide you saw earlier. And so that process itself includes not only clonal isolation, but analysis of the clones, as well as ensuring that the clones are of good quality prior to having those passed on to our researchers for their research, for downstream assay purposes. And so let's spend some time looking at clonal isolation in a little bit more detail. All right, so our single cell isolation process was developed to ensure that the cells we isolate are healthy and they maintain their genetic integrity from that clonal isolation process all the way out to expansion to the final cells that are shipped out. And so let's again go back to our automated workflow, which allows for the isolation and expansion of hundreds of clones in a 384 well plate model that we have shown here. The clones are automatically tracked through imaging. So referring to sort of this part of the workflow right here, the clones are automatically tracked through imaging and cultured cultured with media changes. And so a lot of this ensures that there's very little technical variation. And you'll see that as I speak further down in slides that are to come. So the platform allows for our software to also identify the clones with the highest viability. And these clones can be genotyped in order to identify the preferred clones for end use with the researcher. And so, what does that look like in terms of real data, right? So to me, this is kind of the most interesting part. So the middle part of the slide highlights the workflow that I showed you in the previous slide, where you see the single cell isolation process in a 384 well plate. From there, we're picking those cells with high viability, at the same time genotyping those cells. And when we have that preferred genotype, we expand to get the number of cells per vial that are ideal for end use. But what's important here is the images that I'm showing you below. And so there's a false color for you to be able to see it easier, and also for our software to track it. But the clones with the highest growth rates are hit-picked, as I mentioned, and these clones are further expanded to be vialed. Now, along the top, which is of value to our customers as well, is we're capturing the images, the cells as they're growing at defined time points, and this ensures at the end, when we identify a clone of choice, that it originated from a single cell, right? And so it's part of the variety of QC processes that we have in place, and the automation allows us to do this with ease. So with these two examples, I've taken you through the two capabilities of our module design or our modular design, and highlighted how we ensure efficiency as well as quality in what we produce. And of course, the next part of this is that I would like to spend some time talking about the end-to-end tracking part of our workflow that really allows us to learn from every edit that we do. So spend a little bit of time talking about that. So if you go back to, if you remember from our original slide of, Eclipse capabilities, we do have this value of end-to-end tracking, meaning that, we're literally looking at every single process along our workflow, and we have ways of capturing that data. Now, this end-to-end tracking allows us to learn from every edit that I mentioned that is generated on our platform. As we heard earlier, the platform allows for continuous optimization, Jason highlighted that, and this end-to-end tracking enables that, right? So we use all the previous edits that we have done to best inform and make decisions on future edits. So whether you want to do hundreds of edits with us, as per the case study Jason highlighted, or just a handful, the Eclipse platform really supports and brings value to both. So let me take you through an example here. So these are results from our platform, and what I'm trying to highlight here is how reproducible our platform is, and it minimizes the variability that could theoretically be introduced by other workflows that are not as standardized as what we've created. And so, as a result of this, what we've managed to do is reduce technical variability to the point that we're mostly only having to deal with biological variability. So let me take you through this slide in detail. So what we've done here is we're looking at editing efficiencies, as we have been doing. Across the x-axis here, what you're seeing is basically different guides used in a single cell line. And it happens to be HEC293 in this case. The repeat edits of those same guides, as you can see, cluster together. So for example, this is one guide, this is one guide, and so forth. And so you are seeing that clustering, meaning, the technical variability of repeating that edit is actually pretty reproducible, right? And so what I want to now focus on is what we've highlighted below. So we've taken examples of guides with low editing efficiency, moderate, and high, just to get an understanding of what we're looking at. And what we've done is, what we're showing here is we've at least repeated that edit with the same guide up to 12 times or more. And that's represented by each one of these colored lines as you move upward. And so what you're seeing here now is that for these repeated guide edits, single guide edits, the indel size, which is along here, stays the same. The frequency of the frequency of the different alleles that you would see in a pool, so with a given indel, such as let's say -2 or +4, also stay the same. And that is basically, that is depicted by the fact that all of these guides are pretty much overlapping on top of each other, which is why they're shown at an angle, so you can actually see that a little bit better. And you see this actually across all three guides that we've highlighted here. So what that brings me to is the sentence that's written out here is editing with the same guides in the same context, which is our platform and our workflow, produces the same indel patterns at the same rate. So same indel patterns across this way at the same rates, AKA the frequency. So this shows that our process control minimizes technical variability, which allows us to be more predictable when it comes to the biological aspects that we are dealing with through the workflow, meaning the cell, et cetera, right? So what does that mean? So this predictability comes from the closed loop learning that's specific to our modular workflow. So this is another slide that Jason highlighted earlier. So this means that by integrating our predictive model into our platform, we have built a system that learns about the biology and the technical factors that matter to editing using our workflow. In this way, we can get better over time. And Jason alluded to that as well, right? So that is the graph that's shown here. With our closed loop modeling, we get better over time, right? And while there is no real improvement when using static best practices for editing that researchers commonly use that apply typically around guide design, we're looking at a ton of parameters. And in that way, we can actually get better over time because we're collecting all that data and improving every time we do an edit. So what does that mean for you, right? As someone who's trying to order an engineered cell or get a cell model to study your disease or your gene, it means that your edits run through a workflow that is better at predicting editing outcomes and driving optimization, again, as Jason mentioned earlier in the talk. So ultimately, these benefits can be passed on to you as they come from us for the engineered cells that you order. And so that kind of wraps up the section on our Eclipse capabilities and highlighting the parts of our platform that bring benefits to you as a scientist. What I would like to spend the next couple of minutes is kind of going over or highlighting a pretty amazing effort that we've been absolutely fortunate to be part of to generate cell models for neuroscientists. And so this is, really highlighting our platform, the work that the cells that we've generated through our platform and kind of the value they bring and sort of how this can apply to your day-to-day research, right? So Alzheimer's disease and related dementias, so ADRD, are neurodegenerative disorders affecting more than 6 million people in the United States. Heroic efforts have been undertaken by the research community to develop these disease-modifying therapies for ADRD, right? With even recent news that came out just this week or late last week. Unfortunately, these endeavors, have yielded multiple failed clinical trials, leaving patients with few treatment options. And so what it really comes down to is that our limited understanding of the underlying mechanisms of disease initiation and progression continue to sort of impact the development of these effective therapeutics. So really kind of slowing down that development of effective therapeutics, right? So library of clinically relevant disease models that offer insights into functional linkages between genes implicated in Alzheimer's would expedite the research in this field, and it would also allow for the development of treatments as well, right? And so this amazing effort that we were fortunate to be a part of is called the Inducible Pluripotent Stem Cell Neurodegeneration Initiative, or the INDI project, and it was initiated by the NIH, and it aims to sort of address this gap that I just highlighted to you above. So the INDI is the largest ever iPSC genome engineering project that will model over 100 mutations associated with Alzheimer's disease and related dementias. And it will do so in isogenic iPSC lines. So this would result in cell lines and phenotypic data sets that will literally be broadly shared with the global research community, and therefore hope to sort of expedite the research in this field and also expedite the development of treatments for ADRD. So what was our sort of role in that, right? And so our platform enabled the high throughput of editing of iPSCs. And so using our platform, we were able to generate 264 clones, right, to be exact, if you will. But really, we generated homozygous and heterozygous variants across 22 gene targets. And we did this in just a few months, with the majority of the work being done within about 5 months. And so the ability to process in this parallel way and at scale allowed us to generate these clones with a fairly rapid turnaround time, and therefore potentially accelerating disease research projects. I think I saw Bill messaging us there, so I know Bill's on there with us. We've got you in a quote, Bill, and we thank you for all your support in all of this, and all the push you've had for us, and the support we felt for our platform through your kind words. And so, the engineered cell models that we're producing, like the ones that we generated for the INDI project, people have been working on these, as you can imagine. What we're trying to bring is that optimization and that scalability to it, but it can actually be used in a variety of ways. So from, the hundreds of edits that people are studying to the individual edits that you're interested in, these are some of the papers that we pulled out, specifically in neuroscience, where knockouts, CRISPR-enabled knockouts, were used in studying various aspects of either gene function or pathway analysis or therapeutic development or disease research. And so just a quick highlight in the top left corner, Fragile X, FMR1 implicated in Fragile X was knocked out in human PSCs to generate a model for Fragile X, and it allows researchers to study the function of that gene better, right? And then in your top right corner, what you're seeing is a pathway analysis of a E3 ubiquitin ligase, which has a role in development. Specifically, they focused on neurodevelopment, and what they studied was they used a quantitative proteomic analysis using mass spec to see what the differences between expression patterns of a knockout when Col9 was knocked out in human PSCs and NPCs versus not knocked out, right? So in the wild type. And so this identified various components of metabolic, ubiquitin degradation, and transcriptional regulation pathways. They were disrupted by knocking out Col9. Additionally, there's an example of using CRISPR-Cas9 for therapeutic development in Friedreich's ataxia, where an expansion of GA repeats in intron 1 of the gene causes disease. And so in knocking out those GA repeats, you're creating a solution potentially to a treatment or a treatment process for that disease. And then finally, under kind of the disease research bucket, ALS patients have a hallmark of neuronal cytoplasmic inclusions that are rich in an RNA binding protein known as TDP43. And what they studied here is really looking at what happens when you knock out various portions of that binding protein, and how does that ameliorate the disease and the phenotypic pattern seen with ALS. And so really what we're showing here is there's a variety of ways you can take your cell models, you can of the engineered cells that we're pulling off our platform and carry down downstream assays and research in different ways. So that brings me to sort of the conclusion of what I wanted to talk about today, and I'll pass it back on to Kevin.
Okay, great. Thank you, Grace, for doing that. If it's okay, you can either take us through the rest of the webinar if you like. So hopefully we've been able to show you that our industrialized CRISPR platforms are a great way in which we're trying to apply engineering principles into some of these workflows. It's not an easy task to complete some of these biological workflows and utilize some of these techniques and culture many different types of cells at scale as well. So, it's been a really large amount of effort by the team of engineers and scientists at Synthego to try and make this a reality. Okay, so if you're interested in learning some more about our industrialized CRISPR platforms, please check out the link we've got here at synthego.com/platforms, where you can learn more about the technologies that we're utilizing to help to bring CRISPR genome engineering into everyone's lab in a really democratized way, and ways in which we can help to push your research forward by essentially taking advantage of what we built here. Some useful resources as well. If you're interested, please check out the links here on the bottom of the screen. Okay, so thank you very much for tuning in and listening to how we're hoping to utilize our Synthego Eclipse platform to help forward your own research. And again, we'll be sending a copy out of this webinar to you, a link to view the recording, and also a survey as well. So please go to synthego.com/platforms if you'd like to learn more. And I just wanted to say thank you again to our speakers today, Jason and Grace, for their presentations. So thank you, everybody.